Human milk oligosaccharides and allergic sensitization. What is the current evidence? Karine Blanchard, PhD, Nestlé Research, Switzerland, talked about this topic at the NNI Online Symposium, which was scheduled at the 6th World Congress of Pediatric Gastroenterology, Hepatology and Nutrition. Eczema and food allergy in children are rapidly increasing in the last 2 decades worldwide. Does early life set the trajectory? Gut microbiota development in early life seems to play a role in appropriate immune development. In addition, does breast milk, or more specifically HMOs, play a role in allergic sensitization? Growing evidence from observational studies point towards the role of some HMOs in allergy development. High levels of 2-linked fucosylated HMOs in breast milk seem to be associated with a lower risk of IgE allergy, especially IgE eczema in caesarean section-born infants with hereditary allergy risk at 2 years of age. A further observational study shows that only a specific intermediate level of 2FL in breast milk is associated with a reduced risk of allergic sensitization. Also, FUT3 gene polymorphism in infants seems to be related to the development of allergy and/or sensitization in infants. Further observational studies indicate that not a single HMO but HMO profiles are related to a lower risk of cow's milk protein allergy through 18 months of age or to food sensitization at 1 year of age. In preclinical studies, 2FL or 6SL protects against food allergy symptoms as shown by a reduced diarrhea score. In a murine model, 3SL protects against allergic symptoms and sensitization. Interestingly, a blend of 2FL and LNNT prevent allergic sensitization at an intermediate level of 5%, while this effect is lost at a higher dose. Specific immunoglobulin G1 levels are significantly decreased, and T-regulatory lymphocytes are increased in mesenteric lymph nodes in the presence of a mix of 5% of 2FL and LNNT. Further results from experimental studies show that HMO and 2FL-related microbial metabolites protect against inflammatory-induced gut barrier dysfunction. 2FL and, to a lesser extent, LNT significantly decrease epithelial barrier permeability after an inflammatory challenge, as evidenced by a significant decrease in translocation of FD4 from apical to basolateral compartment compared with non-treated control cells. Similarly, 2FL-related microbial metabolites also decrease epithelial barrier permeability compared with control after inflammatory challenge. In conclusion, HMOs, 2FL and LNNT, decrease allergic sensitization. Regulatory T cells modulation may explain the beneficial effect of HMOs on sensitization. The beneficial effect may also be linked to improved intestinal barrier function. Specific doses of HMO are associated with lower allergic sensitization. Polymorphism in FUT genes needs to be investigated to confound the association.